I'm gonna just jump into the talk directly. Thanks to the previous speakers, it covers the basic introductory stories in my talk. So in this talk, I'm gonna present the actual work to using a combined MR spectroscopy and fMRI, as well as fMRI guided TMS to investigate the effect of TMS targeting on the semantic regions. So again, here I'm going to focus on the TBS protocols. The previous speaker explained that. So we use TMS to modulate the cortical excitability, leading to the behavioral and cognitive changes. And then, for example, using a facilitatory protocol called ITBS, we expected to see the increased cortical excitability, whereas the uninhibitory protocol called CTBS, we would see decreased cortical excitability, mainly in the motor cortex. So from these initial findings, later the same Professor John Ross's group conducted exactly same study with a much larger sample size. And then they found there's no protocol-specific effect using TBS technique. So in this figure shows the individual responsiveness in the motor cortex on the motor evoked potentials. And then here in the black circle represented the group average result. So as you see, there is no protocol-specific effect found across the 52 participants. So they look into the TMS responsiveness at the individual level in this sample, and they found that only 25% of the healthy participants showed the expected protocol-specific effect. And so basically, 25% of the participants basically showed inhibitory effect after CTBS and then facilitatory effect after ITBS in the motor cortex. And then the rest of them are classified as non-responders. There have been many studies looking at this inter-individual variability of TMS responsiveness, and then overall the studies suggested that about 50 to 70% of healthy participants either did not respond or responded in an unexpected manner. Basically, they are non-responders. So this is one major challenge in TMS studies. So that's why we need to understand what determines this TMS responsiveness. Again, this figure, this chart summarized the known contributing factors for the TMS responsiveness. Among them, here we are going to focus on the more intrinsic neurobiological factors, starting with genetics. So there are a few studies look at the genetic variant in relation to the TMS responsiveness. So in this study, particularly looked at the common polymorphism in the brain-deprived neurotrophic factor, which modulate the long-term potentiation, long-term depressions. And they showed that TBS response in the motor cortex was associated with the polymorphism of the BDNF gene that each participant carries. So basically, there are some participants with viral carrier. They are responders. They show the inhibitory effect after CTBS and facilitatory effect after ITBS. Then non-carriers showed reduced or absent TBS effect. So this study basically suggested that some of us can be responders and the others might not. Then another pre-interventional factors, which is a synaptic history. And more recent big TMS data collaboration study demonstrated that baseline MEP amplitude in the motor cortex significantly predicted the response to TMS. So all these studies suggest that the pre-interventional features such as genetics and the synaptic history in the healthy population can be crucial to predict the after effect of TMS protocols. So in this study, we investigated this inter-individual variability of TBS responsiveness, focusing on the higher human cognitive function, mainly semantic memory, which is my topic, using a combined MS spectroscopy, fMRI, and fMRI-guided CTBS. So converging evidence suggested that the anterior temporal lobe is the hub region for semantic memory. So the anterior temporal lobe is our target site for the stimulation. And previously, we showed that GABA concentrations in the anterior temporal lobe was associated with the semantic memory function. So in this study, we specifically hypothesized that the pre-interventional neurochemical features of anterior temporal lobe, including GABA and glutamate concentrations, might be associated with CTBS responsiveness on semantic processing. So we recruited healthy, young, native English speakers. So in their first visit, we obtained MS spectroscopy from the left anterior temporal lobe and then occipital cortex as a control site. So from those two regions, we quantified the level of GABA and then glutamate concentrations, which is a GRX indicator. In the following fMRI session, participants were asked to perform a semantic association task called Kamel and Cocktail's test and a pattern matching task as a control task. So in the semantic association task, they saw three pictures on the screen. The top one is their target picture. They had to choose which bottom item was more meaningfully associated to the target picture. And in the control task, they saw three meaningless patterns. Again, the top pattern is their target, and they had to identify which bottom pattern was identical to the target. So after the initial baseline imaging session, using fMRI, we decided individual customized target site, so-called functional localization for the ATL stimulations. So here, each red dot indicates the individual peak coordinate within the anterior temporal lobe showed greatest activations during semantic processing. Then, using CTBS, we stimulated left ATL and then occipital pole as a control site. And then participants perform the same semantic and control task before and after the stimulation. So let's look at their behavior changes. So again, each red dot indicates the individual semantic task performance. So after ATL stimulations, we observed that there are participants who showed expected inhibitory CTBS effect, showing decreased accuracy in semantic task, and we define them as responders. And the rest of participants did not show the expected inhibitory effect. So we have 20 participants. Overall, we have 12 participants who were classified as responders and 8 participants who were defined as non-responders, which means about 60% of our sample was responders, which is in the line of previous motor studies. And as a group level, the black diamond represent the average group result. We did not find a significant effect. And in the control stimulation, we did not observe any of the stimulation effect. So we grouped our sample as responders and non-responders and look at their semantic task performance. So responders showed decreased semantic accuracy after the ATL stimulation compared to their baseline as well as compared to the control stimulation. However, non-responders showed increased accuracy compared to the baseline, but there was no significant differences in comparison with control stimulations. That is, responders showed a task-specific inhibitory ATL stimulation effect in semantic performance, whereas non-responders did not. And there was no other significant effect found in control task and reaction time. So our behavioral result demonstrated that given in the small sample size, we observed there are responders and non-responders in semantic memory function. Then we explored the relationship between the neurochemical features of the anterior temporal lobe and TMS responsiveness. So this figure plotted the correlation between the excitation inhibition balance in the anterior temporal lobe represented as glutamate GABA ratio, and then their semantic performance changes. We found there is a significant positive correlations between that, which means participants with relatively lower excitation inhibition balance in the anterior temporal lobe, they showed stronger inhibitory effect, and they are classified as responders, colored in red, and then the others are non-responders. So we plotted the level of EIB between two groups, and we found that non-responders had significantly higher EIB level in the anterior temporal lobe than responders. So this result indicate that responders and non-responders may not be a homogeneous group. So there are two different two different populations. So, and next, we examined the baseline semantic task performance in order to explore whether these neurochemical differences in the anterior temporal lobe would reflected in their semantic behavior at the baseline. And the result demonstrated that non-responders performed the semantic task poorer than responders, even in the baseline. So the colored ones are the baseline performance in the ATL stimulation, and then lined a striped bar graph represented the baseline performance in the control stimulation. So finally, the fMRI demonstrated that first, semantic task significantly increased the activation in the all semantic regions, including the anterior temporal lobe and prefrontal cortex and the temporal parietal regions. To investigate neural differences between responders and non-responders, we compared task-induced regional activity in the anterior temporal lobe, as well as functional connectivity within semantic systems. Then we observed that non-responders had stronger semantic task-induced ATL activity, as well as increased functional connectivity in the semantic network compared to responders. So fMRI result also support the findings in the neurochemical systems in the anterior temporal lobe. So the responders and non-responders featured differently in terms of the neurochemical level and the neural levels. So to summarize all the findings, we shows that there is in the inter-individual variability in response to CTBS over the anterior temporal lobe during semantic processing. Importantly, we demonstrated that the baseline excitation inhibition balance in the anterior temporal lobe predicted individual TMS responsiveness in semantic memory function. So our result suggests that the pre-interventional neurochemical features of a cortical region might contribute to TMS effect by shaping each neural trait as well as related cognitive function. So for the method day, so this type of multimodal approach combining multimodal imaging with brain stimulation can provide a more convergent understanding of the neurochemical mechanisms of cognitive function and its related neuroplasticity. So I'd like to thank you for my collaborators, Matt in Cambridge, and then Steve and Sana in Manchester. Thank you for your attention.